just to say, like at the end of the presentation, I'll put those slides. If you're looking for the links or something, I'll put those slides. I'll make a post on my Twitter account so you can access them afterward. So I'll start with a little introduction on the project. Yes, as we all know, the rapid cost, the diminution of the cost of sequencing widely surpasses the evolution of the Moore's law, meaning that basically we get more and more sequences and less power not enough power to analyze it all. Just to give an example, at the McGill Genome Quebec Innovation Center, we expect this year to sequence roughly 500 trillion bases, which approximately corresponds to 5,000 human genomes at the 30x coverage, which means basically that increasing the output of those sequencers imply a constant increase for more compute power to analyze the data, more storage space to put the data somewhere, and also more people to analyze that data. At the center, we have our own cluster to do this kind of job. It's the problem is even high-performance clusters in-house gradually become insufficient to answer the needs of processing and storage requirements for those data. So the one we have at the center is 1,500 cores, and then we have 3 petabytes of disk and 5 petabytes of tape. And unfortunately, it's already full, and we constantly need to make space for the new data to come in. A bit more macro level, for the anticipated need for the 4 Canadian genome centers, 4 main ones, are displayed here. So basically, we expect the number of necessary core to analyze this data to double every year, and the online and tape storage needs are evolving even more quickly. Good. The reasons for that is well, first of all, there's gradually more sequencing centers starting to appear. There's instrument upgrades, which means the data output is increasing gradually as well. We need gradually we need more advanced analysis strategies to harvest all the potential from the data that we sequence. And finally, the increased demand for informatics infrastructure to use data sets coming not necessarily from what we sequence, but from other data sets produced internationally. So, and because of that, one strategy that we went for was to do some partnerships with Compute Canada. Compute Canada has many high-performance computing sites across Canada. We at the center deal mostly with Guillemien at McGill and Mammoth at the University of Sherbrooke, but there's several others. There's one at UDM, and there's many all across Canada. So if you don't know what Compute Canada is, basically it's a national platform which regroups 6 consortia across Canada. And those consortia are composed of several HPC centers. So as I was saying, Guillemien at McGill, Mammoth at University of Sherbrooke, Brière at University of Montreal, and so on, are part of the consortium called Calcul Québec, which is a member of Compute Canada. But there's others for the east of Canada, for the west of Canada, Ontario, and so on. And the thing is, Compute Canada integration also brings some challenges. First of all, because it's not that easy to move those big amount of data sets, very large data set produced from one site to another. When you use several HPC sites, there's another problem is that the software that you install at one specific location also needs to be installed at all the locations where you analyze your data. And on another level, generally the life science community is not traditionally used to run tools at the command line, such as is required of high-performance computing environments, such as Compute Canada. And it rather often relies on high-level tools and web applications. And that's where GenAP comes in. So GenAP leverages Compute Canada HPC infrastructure, so that's like the main goal, maybe. It was released in February 2015. The goals of the platforms are, well, first, to make bioinformatics analysis more accessible to people who are not specialists about bioinformatics, and also to reduce data processing bottlenecks for people who are already using those infrastructures. So common problems tackled by GenApps. Well, first, avoid having multiple copies, as I was saying, of the software installed at multiple places, and same for the genomic libraries and so on. Simplify the deployment, maintenance, and sharing of data across sites. And same for the bioinformatics analysis pipelines. And finally, simplify the access to Compute Canada resources for life scientists, so like making it easier for people who are not bioinformaticians, maybe more like, I don't know, well, PIs and so on, to be able to analyze their data with some tools. So the three main components of GenAP, well, first of all, there's a portal. So that's the main thing that I'm going to present to you today. But there's also the CVMFS component that I'm going to talk about quickly, and the bioinformatics analysis pipelines, which Louis has presented, has presented at MonBug 2 months ago. So I'm mostly going to talk about the portal, but I'll go around with the other aspects as well. So, well, first, I'll talk about the pipelines and tools which were presented, as I said, 2 months ago. Those tools are already installed at several Compute Canada HPCs, and they're used at multiple institutions. So basically, what GenAP offers on that front is a framework to like object-oriented, written in Python, and which eases the construction of those bioinformatics analysis pipelines, and offers different things such as error recovery and logging and so on. That framework has been used also to implement a couple of pipelines already, so which are also available. I'm talking about DNA-seq, RNA-seq, ChIP-seq, RNA-seq de novo, and so on. We're continuing to develop new pipelines, and those are available online. Actually, the source code for the whole project, the pipeline frameworks and the pipeline implementation is available on the Bitbucket source control repository. It's open source, so you can do whatever you want with it. We have a mailing list, so like if you have questions or something, we can answer them, and an issue tracking systems for hypothetical bugs that could be found. And the good thing is, like, the documentation for that is available in the code as markdowns, which means, like, basically when you consult the on Bitbucket, when you consult the source code, well, you have access to the documentation as well. The next aspect is CVMFS. So CVMFS stands for CERN Virtual Machine File System. It was originally developed for the LHC, the Large Hadron Collider, to distribute code at multiple locations for their VMs. It's designed to be fast, scalable, reliable, and so on. And software can be installed at one location, that's the main concept, and like distributed everywhere. So like the way it works is basically somebody that we we call a librarian, will log into the Stratum 0, which is like the central repositories, if you want, or the central place for the file system, will install some software, and then we'll publish it. And once the software is published, it's being pushed to the different Stratum 1 instances, which are mirrors of this library. And then from there, these Stratum 1 instances will be accessible to some squid caches, which are distributed at different locations, and software will be available this way. So if I'm, for example, if I'm logged into the GenAP, and I want to execute to launch a specific software using specific libraries, I will launch my commands using the software installed in CVMFS. If the software is not available on my local cache, a request will be launched to the squid cache, which is located at proximity. If the squid cache doesn't have the software that I'm looking for, then it's going to interrogate the Stratum 1 to download the necessary software and libraries, and then my commands will be executed. So it's just to say, like, so we have, of course, one Stratum 1, and right now we have one, so we have one Stratum 0, and right now we have one Stratum 1, which is located in Quebec, and we're working to have another Stratum 1 in British Columbia somewhere like this year. And we have a couple of squid caches, like we have one at Sherbrooke, we have one on Abacus, which is the cluster at the Genome Center. We have one on Briaré. We have a couple of ones across Canada, in Vancouver and in Ontario as well. So this is like just the big picture for this. So like this, there's the Stratum 0, which is here. Once you push the code in, it's being distributed on the different Stratum 1s, and then somebody who's, for example, logged on GenAP will launch a command which will fetch the necessary libraries and software and from the local squid, if it's available. If it's not available, it's going to get it from a Stratum 1, and then brought back to the squid, brought back to the local cache, which will execute the software. The third component of the project is the GenAP Portal, which is like the main part of this presentation. There you go. I should have done that from the beginning. So the purpose of the portal is to manage GenAP services and resources. That I'll come back to that soon. So what it does is, well, first it authenticates against the Compute Canada database, which means when you have an account at Compute Canada, you have an account on GenAP. You can log in automatically. You don't need to ask for new access and so on. So the basic setup is when somebody logs, maybe a PI logs into GenAP for the first time, there's a project that gets automatically instantiated, and like everybody who's under his or her allocation at Compute Canada gets automatically an account as well, and will get affected to that project. So like that's the default setup, but the portal, as I will show you, has several tools which allow you to configure these things. So one group can have like several projects with different people working on the same things, and like it's also possible at Compute Canada, for example, if you have some allocation but you have some external collaborators that you would like to work on this project as well, it's possible to ask Compute Canada for them to have an account that you like to kind of sponsor them under your own allocation, you know, and then those collaborators can access GenAP as well and start working with your data if that's what you want. So you can like fine-tune per project. If you have several projects going on, who's going to work on what, who's going to be able to access what. And once you create the project and you configure it, or if you take the default configuration, the last thing that's remaining is to basically instantiate the service. I'll come back on the service. So, well, yeah, so the first and maybe most important service for now is Galaxy. So I don't know if I haven't really prepared a presentation part about Galaxy. I don't know if you're all familiar with what it is, but basically it's this framework which allows you to launch jobs to use bioinformatics analysis tools using a graphical interface instead of doing it from the command line. So what the special flavor of GenAP, of Galaxy for GenAP, is that like we adapted Galaxy to be able to use the Compute Canada HPC allocation to run jobs. So what this means is like if you have a Compute Canada account, you have your space and cores allocation. You can launch jobs. I don't know. You can launch an alignment job or anything, and like this job will run using your Compute Canada allocation instead of, I don't know, like some general pool like would be on the main Galaxy service. So our GenAP offers over 400 tools. We are always open to offer install new tools on request. And one important thing is like each group has their own private instance, if you want it, its own private space in Galaxy, which like prevents, I don't know, like either like for security reasons and for management reasons and those things. So if you create an instance of GenAP for a specific project, only people in this project will be able to access it. Another service that GenAP offers is the Data Hub. Basically what the Data Hub is like at the super macro level, the Data Hub is like just an open space to the web. So like you have a Compute Canada allocation, say at Mammoth, you instantiate a Data Hub, so you reserve a certain portion of your allocation to be a web space. And the reason for that, the main use for that is like there's this tool for the UCC Genome browser called the UCC Track Hubs, where you can produce annotation tracks for your data sets to be visualized in the Genome browser. You can just dump everything that you produce there and that you want the world to access. You can just put it there and like, and then your data becomes accessible. So like basically it's a way for people to share their data. We've got some people using it to put their published materials. So like they produce some data sets, they want to publish a paper. So basically they have a data hub, they put their data there. Now it has a permanent URL, which is not encrypted, but the URL is a bit cryptic to make it so like it's hard to guess for somebody who comes out of nowhere to just accidentally fall on your data, but it is accessible to the world. But so like you can just put your data there and I don't know, send your article to a journal or to collaborators and something who can then access your data, do what you want with it. That's why I put a picture of a Genome browser here, because like this is like just a track hub that I load in UCSC Genome browser. Speaking of which, another service that we offer is we have our own mirror of the UCSC Genome browser. So if you find the main one too slow, well, we have the whole set of annotations for human HG19, HG38, and the mouse MM10. And yeah, that's it. So the full Genome browser is accessible to load your own tracks and so on. So like the permanent URL is here. So the next service I wanted to talk about is the IHEC Data Portal that we developed over the past years. So IHEC stands for the International Human Epigenome Consortium. Basically, it's a consortium regrouping several consortia across different countries who aim to produce what they call full reference epigenomes. What this means is that, like, for a sample or maybe a group or a pool of samples, you're going to have assays on different epigenetic assays, such as RNA-seq, several marks for ChIP-seq, whole genome bisulfite sequencing, and so on. So there's this group, all these groups produce data sets, and like we were looking for a way to merge them together, to offer them in like a kind of directory that people can consult, and that's why we created the IHEC data portal. So the goal is data sets discovery. Basically, if you're working in the field of epigenetics and you want to know what's available from IHEC, you go to the site and then you have these several tools to try to find the data sets that you're looking for. There's over 5,500 data sets right now. I can show you a quick demo. So this is what the portal looks like right now. So well, right now available are 5,667 data sets. Basically, you have tools to select data slices that you want to work on. For example, say I want to visualize all data sets for blood, maybe made in Canada, and let's say in the Blueprint Consortium in Europe. Then if I view the selected available data sets, there's this grid which displays how many data sets per cell type, and per assay type, and then basically you can select the data sets that you want. And once you're ready, you can visualize those data sets in the Genome browser, which will take a little while to do, but what's happening there is like there's a dynamic track hub that's getting created, which shows all data sets for your selection. That's it. So now I'm going to do a little demo of the portal, which is bold, but even more bold is we'll do it on our dev server. So hopefully everything will be fine, but it should be. So this is the main page. If you go to genap.ca, I'll just open it on a bigger page. So, well, there's a couple of information about the project, the different components for GenAP and those things. But like, if I log in, again, I'm using my Compute Canada account and password to connect to the portal. I'd like to note that, because we had that question a couple of times in the past, the login password that I'm using here is not the password that you use to connect, for example, to Mammoth or to Genome specifically. It's the password that you use to log into the Compute Canada CCDB, the Compute Canada portal. So once I do this, I'm brought to this GenAP main menu here, which gives me, well, a couple of news about what's going on in the GenAP project, the applications that I most recently accessed. But maybe I'll start by getting an overview of the projects that I have created. So, well, did I mention it's a test server, so there's a bunch of test projects. But just to say, like, so I have several projects here. Some have applications instantiated, others don't. And then what I can do is, if I open this page for my brand new project, is I can, well, here's a couple of details about the project, and then I can manage, as I was saying, the members who are in this project. I can add new members, say, I don't know if, okay, let's take this Alain and add him to my project. I know him a bit, so I just added him to my project. So like now this guy can log into any application that are part of my brand new project, such as if I instantiate galaxies, if I have data hubs. He can connect and he can use them. And how do I instantiate an application? Well, I go to the application folder, and I click on create an application. So right now there's two choices. We're working on adding more tools as we go on with the project. So well, I already have a Galaxy, but let's say I create another one. Right now, the only GenApp node available is at University of Sherbrooke, but something like by September, normally we should have a new node available at Guillemins. So that's why it's kind of pending. It's coming soon. But right now I'm going to instantiate and applicate the Galaxy at University of Sherbrooke. I'm going to give it a name. And there you go. Okay. So once you create an application, it takes a bit of time for it to be available. Same for the project. So yeah, I didn't show. Oh, there you go. I knew this would happen. You know, but seriously, when you create an application or a project, it takes some time for it to become available, just because like there's quite a lot of work happening in the background. When I instantiate an application, that means instantiating a new virtual machine, giving proper access, running a whole bunch of scripts for this virtual machine to be accessible, and so on. So that's why like you can expect, maybe you'll have to wait like a couple of minutes up to maybe half an hour before having your application accessible. But once it's done, then it's ready to go. And this is what's saying here. I guess I clicked on submit twice. Probably that's why it's installing two Galaxies right now. But I already have one accessible as well, so maybe I'll just try to open it. So if I click on the application, it's going to bring me to this specific instance of Galaxy that I created. So I could give you a little tour of the Galaxy interface and those things, but like, how many people are not familiar with Galaxy? Okay, well, I can explain a bit. Galaxy, as I was saying, offers this whole bunch of tools related to genomics, and like it has features such as so you can load in your data sets, and then you can either just cherry pick the tools that you want to apply to your data set, or there's also this concept of workflows where once you load data sets, you can execute a whole bunch of operations one after the other on your data set until you obtain the result that you want. So in the case of GenAP, there's two ways to add data to your Galaxy Galaxy space. The first way is using the get data upload files option. So sorry. So basically, like you're going to browse and you're going to add manually the files that you want to work in. You can multi-select them and then just click go, and like even if it's like in the order of gigabytes of those things, it's going to load it eventually. It's just that it's through HTTP, so it's not very fast. There's another option you can use, which is when you create an instance of Galaxy on the GenAP node, which is at Mammoth in this case right now. There's a dump directory that gets created. So if you have data sets that were generated elsewhere on the Mammoth cluster or at another cluster, you can just copy or rsync your data in this folder, and then you can go to this instance of Galaxy, and you can load them in through the interface. So you can use this to link file and server to the current history. So right now there's nothing, but if I copied like some BAM files to this directory that I'm talking about, then they're gonna get available here. And what that means, like, you just need to register them in your Galaxy space, and then they become usable in the same way as if you uploaded them through the upload file option. And so that's how you get data in. Now, how do you do something with your data? Well, maybe since I don't have at hand a data set available, I'm just going to download some random, well, not so random, but a data set coming from the UCSC Genome browser. So there's this option here, which allows you to download a whole bunch of publicly available data sets, and then you can do stuff like either just with that, or more interestingly, you can use some tools with your data, with your own data, and using this as a reference as well. So if I click on get output in this case, and then I send the query to Galaxy, then I'm asking to download on the, I think I selected just the exons, I'm not sure, but like I'm downloading the whole set of known genes as a bed file into my Galaxy space. And you were seeing it in yellow, that means like the version is going on, and once the version is finished, it becomes green, and that means my step is finished. And I can use this bed file, which just got downloaded, as an input for another tool. So as I was saying, there's a lot of tools here, such as like genomic assembly tools. There's like different kind of tool for annotations, for RNA-seq, for methylation, and so on. And as I was saying, like if you have specific needs of something that you don't see in the menu there, you can just ask us, and like in the next version we can add them to the list. One thing I want to specify is like inside GenAP we will have like two releases per year of a refreshed version of Galaxy, because Galaxy wasn't, have to mention, it wasn't developed by our group. It was developed by Penn State University, I believe, and some other groups. And we, like, basically what we did is we took that and we integrated it there. They continue working on Galaxy to make it evolve. Like twice a year, we'll take a snapshot of like the most recent version of what they have and offer it as a new version of Galaxy, so that we stay always up to date. So because of that, in those twice yearly releases, if there's extra tools which are not available by default on Galaxy or in our instance, just let us know and we will add. And that's so like I don't think I have much time to cover a lot about Galaxy. We do offer we did offer some workshops about, more specifically about Galaxy at the Genome Center in the past. So if you have any questions about the use of those things, you can ask me after, and I'll be glad to guide you a bit through how it's working. So that's it. Now I explored the Galaxy aspect, and just, do I have a data hub accessible here? I know, I just want to show you one quickly, but it doesn't seem like I have any data hub available under my account. But once that service gets created, there's something a bit similar to what I was talking about in Galaxy. Like, so you instantiate a GenAP Data Hub service. It creates a folder where you can drop your stuff in. You can connect through SFTP to upload your data. And as soon as you put data there, or that you copy it using the command line, your data becomes available. So that's for the project part. Like under my application, you can see like the list of like all the most recent applications that you instantiated. You can get a list of all the multiple Galaxy instances that I instantiated in the past. So yeah, that covers the application part. Like one interesting way I see of using these multiple instances through project is those things. Like we have some PI at the center who have like collaborators both in Canada and outside of Canada, and what that person will do is so create a project, ask those persons, will sponsor these persons to get a Compute Canada account, add them to the project, and then these people will be able to handle the data through our Galaxy instead of just sending them a bunch of files which they will not necessarily know what to do with, because Galaxy offers like all those visualization tools and those things as well. So that's it. Those are like the portal's main mission is that it's like you create your project, you create your application, and then you use it to access those data. I can show you maybe there. Well, this is just like some statistics, like showing the usage of the pipelines at the Genome Center from different clusters, and that's pretty much what you can do through the portal. So that's it. I'll continue. So the future plans for GenAP. Right now we're working on better robustness, better reliability. People are giving us comments on how to make things like even better than what they are right now. So we're working on the interface to make it good. One of the big plans that we're working on right now is bringing CVMFS into Galaxy, which means basically that like all the tools which are being used by the Galaxy instances, like I've shown you, will be served by CVMFS, which will make it easy to create new GenAP nodes in the future. We want, of course, to deploy the GenAP nodes, more GenAP nodes at more hosts, and then we have all those interesting plans about data management across sites. Say, like I have data that I analyze with Galaxy at one site, and I want to merge it with the data set which is at another site. Then we are in discussions with the Globus group to make tools to make that easier. And we're looking to, of course, implement the other services that are tailored for genomics. So how do you get started? Just to recap, basically you create an account with Compute Canada if you don't have one already. You log into the GenAP portal and you instantiate the desired services. How do you get an account with Compute Canada? So like I put all the URLs here, so I invite you to check the slide after if you want. First of all, you create an account with the Compute Canada website, which is right here. And then, since GenAP's nodes right now are part of are at 2 HPCs, which are part of Calcul Québec, then you need to create a Calcul Québec account. You do that through the Compute Canada portal. So basically, once you have your account at Compute Canada, you log into their portal and you click on Apply for a consortium account. You click Calcul Québec and you make the request. They're going to do that for you. And after that, you need to go to the Calcul Québec portal and ask under the My Profile which specific HPC you want to access. In this case, Montreal. So these are the funding and partners for the GenAP project. GenAP and Genome Québec, Canadian funder, and Genome Québec funding as well for our first part of the GenAP project that we released. Partners are with Sherbrooke University, McGill, Compute Canada, and Sargan Innovation.ca. Here's the team that mostly work on different components of GenAP. There's lots of people at McGill University and others at at the University of Sherbrooke. And there you go, like that covers the topics that I wanted to cover to talk about today. Thank you very much.
Any questions?
You haven't mentioned what is involved in creating a new application. If you have a group that has some mathematics application that they want to have available to the app, what is the kind of work that is?
I mean, if you develop a new, if you develop software that you want to have,
Suppose we have our own portal that we would visually instantiate inside the app.
okay. When you talk about, so maybe you develop some web application that does something. That's not really the goal of GenAP right now. Like if you have a tool that you would like to integrate as a service so that everybody can instantiate and start using, then I invite you to communicate with us and just tell us just introduce us to your tool and those things, and then we'll discuss to add this, and this should be doable. Is that clear?
But it's a virtual machine that needs to be prepared?
Yes, exactly. So like
it's a complete operating system.
Yeah. So just maybe I didn't make this clear enough, but like everything that I presented, like whether it's Galaxy, the data hubs, the IRCM data portal, USCSC Genome browser, and all the tools which are offered as services of GenAP are all contained, self-contained virtual machines with their own OS and those things. So we have these servers which are hosting all of those virtual machines with all their various kinds of services. So like in a case like you're talking about, there's basically two choices. Like if you created something that like Galaxy styles that you want to have, you want groups to be able to instantiate, then we need to do some particular setup for that. But like what we do most of the time is we are collaborating with Pierre Saint-Jacques at the University of Sherbrooke. They're developing a couple of web tools like that we offer in one contained virtual machine that like doesn't get multiplied or something, but basically it's like a server for your web apps. Yes. Does that answer well? Here.
Yeah, when you were doing the test, you click on pipelines there.
There you go. Okay.
What's that? Is that the Python pipelines you run manually, or these are pipelines inside of the Galaxy?
Okay, those are pipelines that you run manually. So basically, it's like we just to know whether those pipelines are being used or not, we put this optional feature where like when people use our pipelines, we just get a hit to know that it's being.
That pipeline was run. Okay.
Yeah, exactly.
When I was using that, because I used the GenAP, and that part I never understood what it was doing for me.
Okay, actually, we were maybe it's a bit misleading since it's actually concerning the pipelines which are run at the command line. It's maybe less relevant for the portal, so we were thinking maybe we should leave it there.
Analysis pipeline ran by.
That's a good idea. Yeah, it would make things clearer. Thanks.
So I was running stuff and nothing was doing anything there.
Okay. Thank you.
Is there any plan for using GenAP resource to install the solution?
So yes, for GenAP we have I know the pipelines are installed at BIA. I know that there's now for CVMFS there's a squid cache at BIA. So meaning like at the command line you can use the pipelines developed as part of GenAP on the BIA HPC. For the GenAP node offering Galaxy and the data hub, like yes, we want to reach that. As a short-term plan, we would like to make, like we're doing things at McGill just to make sure, like fine-tune everything to make sure that it's very really portable, and like then we're open to in any HPCs which would like to become a GenAP host, a GenAP node, we would be glad to go with that. So I would see that as happening not that much like until autumn maybe, but after that, once we're set up at Genome, then we'll be willing to.
So when you launch an application inside Linux, it will be dispatched as job on the cluster outside of the virtual machine.
Exactly. Yes. So yeah, nothing runs on the virtual machine itself. So and yeah, I didn't mention that, but like there's basically two types of jobs from what you run in Galaxy. So some are more like computationally intensive. You know, like I don't know if you run Top Hat or Cufflinks or something to quantify your transcripts. Those are jobs which like take a while to execute. Those are launched as normal jobs on the HPC. But like we also have like a couple of serial nodes for serial jobs. So like there's a special queue for like jobs which are quick to execute. So I don't know, just a file parser or something like that that takes 10 minutes to run. That wouldn't be launched on the main queue because sometimes you have to wait like a day or two before it starts, right? For a job that takes 5 minutes, it's not really worth it. So for all the quick running jobs, we have special queues. We have two nodes entirely dedicated for that, of 24 core each. So like those are doing the fast running jobs, and there's a regular queue for the bigger jobs. Point is because I'm that's because I have the administrative right on the portal. You will not see that. So yeah, there's no. But there, you have an option to delete though. So like if you instantiated like 2, 3, 4, 5 galaxies and you realize that they're useless or something, it allows you to do a bit of housekeeping because we don't have that yet, but soon we will put a limit on how many Galaxy instances one group can instantiate, just because like it does take a substantial amount of memory and those things. So like just to make things like to keep things clean, we will.
Is it better to just stop? So that when it's running, if I'm not using it for a couple of days, it's better to just stop.
Well, normally it doesn't really matter. Yeah, it's just like if you have 50 of them, you just use one. Yeah, I would recommend to stop the other ones, but.
And when you're not using it, it's in running mode. Does it take any of your points of?
No, it doesn't. If you don't launch jobs, it doesn't take anything from your allocation. Just like if it generates files, well, of course the files which sit there, they take you use your space, but it doesn't take your computer allocation.
Do you have any feeling for like how many users you have, and maybe how many? I mean, this is a tough question, but how many labs are using it that don't have sort of the computational background? Is that something you guys have a feeling for?
Yeah. Well, I can show you this. Well, that's we just added this monitoring tool, so to give us an idea of like that's the test server, so it's not really. I will need to log into the main portal, but well, actually, just give me a second. Okay, it's to you, like we released it yesterday. Okay, so I know we got a couple of dozens of users for Galaxy and those things, but I don't have a precise number on how many different labs are using it.
I mean, I'm just more curious as to, do non-computational or non-computer scientists have any aptitude for the tool? Like, is it something that you can approach biologists with?
That's our goal for the Galaxy component. Like, so we organized with Genome Quebec a Galaxy workshop last December, where like the target was like beginners for computational biology and those things. So people who don't know how to start something from the command line and those things. And yeah, we got good feedback from there. So I think those tools are really tailored for this kind of users.
I can comment on that as well, on the same question. Coming from previously a biological background, galaxies are really popular for that, because you don't need to know any command line, anything to have the tools. You just need to know what the tool does, basically. So you need to know the concept of, like, I want to align my reads or stuff like that, and it's just a click, basically, and everything is done, right? And with the default parameters and stuff like that. So I think it will be really useful for people that don't have any.
How were you exposed to it, if you don't mind me asking? If you didn't come from a computational background, how did you encounter this?
Well, I did a master's in microbiology, and I was using Galaxy on the public servers to do some transcriptomic analysis, and I really liked it. So then I did some more study into bioinformatics, and then I got information from a job from that.
So you're converting them.
Yeah. But yeah, Galaxy was really easy to use, like no computational background at all.
I hope we still have jobs just get produced. So you have slacks and vertices. There's no fork this time. There's